All right, let's go ahead and get started. So the only new announcement, and I'm sure you're all aware that there will be no class sections or office hours next Thursday and Friday because those are university holidays. There will be no new homework posted next week. So the homework that's posted tonight, you'll have the opportunity to go through this Friday or Monday, depending on when your discussion section is. Any questions? All right, so we began the last class going over this this last RNA-mediated genome editing process called CRISPR. And so let's start it with a clicker question to see how much you all remember of that. The internet didn't go out, did it? All right. Oh. Uh-oh. All right, it's telling me there's no base. That is not good. Let's... Sorry. We'll try to fix this. Quick, click in. So it's turn your clicker off and turn it back on. No? Everyone who has a manual clicker, is it not working? Is anyone's working, manual clicker? Because it's saying I have a base, it's saying this is working, but the phones are working? Hey. All right. If we don't get it working by the next clicker question in the class and you still need a clicker point, you can email me later and I will give you the point for being here in class and clicking in. If you already have 10 points, keep clicking, but you don't need to email me. You already have the maximum amount. So hopefully it will work for the next clicker question. All right. So did everyone click in who was able to click in? I'm sorry about these technical problems. All right, so let's see what the answer is for that. Oh, this does not bode well. All right, so the vast majority of you did choose B. So who wants to tell me why they chose B or perhaps a different answer? Nick. Right, so Cas9 is indeed an endonuclease, but it's what causes the cleavage through the double-stranded DNA that you targeted, and the repair is done by the cellular factors, either for end joining or homologous recombination. So A is not correct. B, why did you like B?
Well, then you could just, from the answer in A, C because RNA doesn't act as a backbone catalyzed in cleavage. That's what the endonuclease does. And it's a double-stranded cut, so that would also make D wrong. But the small guided RNA does base pair to the DNA to signal where the cleavage is.
Exactly. That's how we get the specificity in our CRISPR experiment. It is that small guide RNA that is searching for a sequence in the DNA to which it can base pair. It is in a complex with the Cas9 endonuclease, which then will cut through the double-stranded DNA. All right, and so just as a reminder of how CRISPR works. So, as we said, we need the small guide RNA. That's what's going to give us our specificity for where in the genome to direct the Cas9 endonuclease protein to cleave the double-stranded DNA. We know once we get a break, a double-stranded break in DNA, there are two choices for how to repair that. One is just through non-homologous end joining. So suppose you just want a collection of mutations in a specific gene you want to study. You can just direct cleavage. End joining will result in errors in the repair mechanism, so you'll get your collection of mutants. If you want a specific change in your DNA, then you're going to depend on homologous recombination. And for that, you would also need to include a repair template. So when you do your CRISPR experiment and you introduce the guide RNA and the endonuclease to the cells, you also have to add your repair template that will have the sequence you want incorporated, and flanking that sequence just needs to be homologous sequence. It just needs to match the positions where you directed the cleavage. Any questions about CRISPR? All right, so then we went on and talked about creating recombinant DNA, which is just DNA from two different sources combined together. And typically, we take advantage of these plasmid vectors that will be able to be replicated in bacteria cells. And for efficient cloning of our DNA fragment insert of interest, these vectors typically have three important elements that will help us. First of all, if we want this vector to be copied inside the bacteria cells, what kind of sequence do we need in the vector? The what? Not the open frame sequence, but so for copying or replication, what do we need? The origin of replication, ORI. So we're going to need a sequence that the bacteria proteins will recognize to copy. We then usually have a region that has multiple restriction enzyme sites. We call this the multiple cloning site or polylinker site. And that just allows us to be able to cut open the vector at one position to open it up so that we can clone our DNA fragment into it. And then once we have cloned our fragment into the plasmid DNA using ligase to ligate the insert into the plasmid, we then need to get this recombinant DNA into bacteria cells. We sometimes call that transforming the cells. It's basically getting the DNA inside the bacteria cell. And the thing is the methods we use to transform the recombinant DNA into the bacteria cells usually wind up with only a small portion of the bacteria actually picking up our recombinant DNA. And that's where our third element in the vector comes into play. We need to have a selection marker gene. And so this is a gene that will express a protein in those bacteria cells. So only the bacteria that have picked up your recombinant DNA will be able to survive. For example, if it is a gene encoding ampicillin resistance, you would grow your bacteria on the ampicillin antibiotic. All the bacteria would die except for those that have that resistance gene. And so those would grow up. Every time they divide, the cellular machinery would then copy your recombinant DNA, and you can then isolate that DNA, sequence it, make sure it is actually the recombinant DNA you intended to make. And those are the basic steps of how we would create and amplify our recombinant DNA. Any questions about that? Oh, no. All right, so we're going to do a clicker question. And now probably nobody can click in because it's telling me no base again. Let's see if we can change that. You can start reading in the meantime. Let's see. Weird. It like doesn't even see it. Hey. Oh. No. Let me try one more thing and then. it work? For everyone? Okay, good. So you'll get full credit for today as long as you click in for one of the questions. So click in. So I will give you a hint. The sequences of the restriction enzymes that are our options, none of them are found in this insert sequence here. All right, how about 10 more seconds? All right, final answers? Okay, lots of options here. All right, let's walk through this and make sure we understand the thought process behind how you would design this. All right, so what I'm asking you to do is I want you to amplify the entire promoter sequence. So this is small for a promoter sequence, but you want this entire sequence to be amplified. Here's your Tata box, and you want to insert it upstream of the GFP coding sequence in the vector. So this is a promoter. We want to drive GFP expression. Do we need directional cloning? Does it matter which orientation it goes in? Absolutely, right? So if this Tata sequence is over here, it's going to direct transcription in the opposite orientation. So we want to do directional cloning. When we're thinking about directional cloning, we usually want two different restriction enzymes on either side, so the puzzle pieces will only fit together in one orientation. So that's one thing to think about. What's another thing I should be thinking about looking at this vector I have? You don't want to have BAM be one of your choices, because what's going to happen, you cut your vector with BAM H1. Yeah, you'll cut in this upstream region, but you're also going to cut right in the middle of the GFP, and so this fragment is going to be lost. You're not even going to be able to encode GFP anymore. So BAM H1 is out of our choices. All right, we want to do directional cloning, so that means on this end, the part that's furthest away from the +1 site, do we want to have not 1 or echo R1? Not 1, exactly. So we're going to put not 1 over here, and we'll put echo 1 over here. So we know which restriction enzymes we want to use, and what is the trick we learned for our PCR primers? Where can we put those restriction sequences in our primers? 5' ends or 3' ends? 5' ends, right? We need the 3' ends to be able to perfectly base pair with the template so that you can get synthesis. All right, so we all remember how to design basic PCR primers, right? And one easy thing to think about is just make your template double-stranded, denature it, and then make sure that you will have sequences with the 3' end perfectly base paired facing the other sequence going in this direction. So you're going to need to have these sequences at the 3' ends of your primer. And so if you look at your options down here, all right, that already narrows it down to a few that have those sequences. And then we said, so the blue sequence over here, is this going to get not 1 sequence or echo R1 sequence? Not 1, right. We said we wanted not 1 to be down here, so we're going to put the echo R1 over here. And so if you draw it out, you can see that here's a sequence that perfectly base pairs. here is a NOT1 sequence. You just write it exactly as it's written, 5' to 3'. Write that exact sequence onto your normal primer. Over here, you can put echo R1. Again, reading it just 5' to 3'. So 5' is GAATTC, and then the rest of the primer. And then during your PCR reaction, of course, you will get copying, and through multiple rounds, because the new piece of DNA that has this primer sequence on it becomes the template for the next round, many rounds of PCR, what you're going to end up with then are, oh, I didn't draw it out, sorry. You will get the insert amplified with these restriction sites on either side. And so I didn't ask anyone what their favorite answer was, but the B is actually the only one that ends up having sequences that will base pair to the template correctly, and then have your NOT1 sequence over here and your echo R1 sequence at the 5' end on this primer. Question?
Why do you put like NOT1 5' side?
So why did we do that? So let's go back and look at so here is the gene we want to express, meaning we want transcription to go in this direction to make this GFP messenger RNA. This is our template, or is our promoter sequence. I told you here's the Tata box right here. You can see this is about 25 to 35 bases upstream of what will be your +1. So you want this to go in the orientation where this end is furthest from the start +1 site and this site is closest, right? So which restriction enzyme, we already said we couldn't use BAMH1, which one is furthest from the +1 site? NOT1, right? So NOT1's going to go over here, echo R1's going to go over there. Does that make sense to everyone? And I'll, of course, include these additional slides in the when I post tonight. All right, so then we said once we create our recombinant DNA, we would want to sequence it to verify it. And if we just have one piece of DNA and we want to sequence it, we send it for what we call Sanger sequencing. We send this out to companies now. It gives us back the sequence. It also gives us a trace so we can verify whether the nucleotide reads are correct. But then we went in and started to talk about, we also have a relatively new sequencing ability, which is considered to be next generation sequencing or high throughput sequencing or parallel sequencing. Those all mean the same thing, because what it allows you to do is sequence millions of strands of DNA at the same time. And we went through the steps of how you can create a cluster of amplified molecules of DNA and then read step by step depending on the fluorescent nucleotide that is added. I told you don't worry about the details of that sequencing method. It's just one version of how we do next generation sequencing. But instead appreciate that this method allows us to sequence an entire genome because all the little pieces of strands are being sequenced at the same time. And then we left off in class with this question of, well, why would we want to sequence a human genome? And there's a lot of very important practical applications to this. Excuse me. And so one example is that sometimes there are infants that present in the neonatal intensive care unit. You do a screen for a series of known genetic diseases. Nothing comes up, but the baby is not thriving. So what is the problem? And so this sequencing method, which actually was partly pioneered here at Rady Hospital, said, well, let's just go in, sequence their whole genome. Maybe there is a mutation somewhere in a gene we know something about, and that might help us identify the cause of these symptoms, and better yet, offer a treatment for it. And this is really important because these days we can sequence a genome in 24 hours time. That's a very quick turnover to sequence and then read that genome and look for mutations. Sometimes, depending on the type of mutation, if you start treatment right away, you avoid neurological damage, organ damage. In some of the successful cases where they've used this, they identified that the baby wasn't doing well because it couldn't metabolize a single amino acid. All you have to do is change the diet of the baby, and then they thrive and do better. And so this is a really powerful method that is now been used thousands and thousands of times, and so it's really shows the power of being able to sequence quickly an entire genome. Another area where sequencing entire genomes has become really important is for cancer and tumors. So as we'll talk about in the last lecture, a cancer development, cancer is basically your own normal cell picking up a variety of mutations and then dividing out of control. Knowing what those specific mutations are can really help you determine the treatment for that particular cancer patient. Some patients, if you have certain mutations, there's certain chemotherapies that are just never going to work for you. So there's no point in giving these harsh chemicals to the patient if they're just not going to work because of the specific types of mutations. Tumors have a variety of different types of mutations. Another recent use of DNA sequencing was able to diagnose pathogen infections. And so there were some patients who were having some neurological symptoms, and so they isolated the spinal fluid, did sequencing to ask, is there anything in there besides human DNA? And in the case that I'm talking about, they actually found meningitis. Meningitis is a fungus that can be treated once you know what it is, and so very powerful method. We also have a newer method called Nanopore. This allows us to do direct sequencing. We don't have to fragment the DNA. We actually can feed the DNA into these flow cells, and it will give us the read. And so this is a very powerful method, and the size here, they actually are only about this big, and they're relatively cheap compared to the other type of sequencing. My lab has 2 of these Nanopore sequencers. There's pros and cons to doing this type of sequencing versus the high throughput sequence I talked about before. And then there's also, a big initiative to get the sequences of as many different types of samples and people as possible, because when we are comparing these tumor genomes or these neonatal genomes to the human genome, we call this the reference genome. This is just the DNA that was originally sequenced from volunteers who were of European descent in northern New York. So, of course, all of our genomes differ, and the bigger collection we have, the pan-genome we have, will help us understand better how differences, how variants can relate to different characteristics, as well as when a variant might actually be disease-causing. All right, so finally, let's talk about what we learned about making recombinant DNA actually is extremely useful for getting bacteria or other single-cell systems to act as little factories to pump out medically relevant proteins. And so the example here is insulin. This is the first example of a protein we were able to engineer bacteria to make for diabetic humans to use as a therapeutic. And it actually takes advantage of knowing some basic information about how gene expression is regulated. So I'm sure you all remember the Lac Operon. Remember when we talked about transcription in bacteria, and we said we're only going to talk about one example of regulated expression? Well, it turns out that understanding this system is very powerful for making these protein-producing bacteria factories. So you'll remember that the natural LAC-Z promoter in bacteria has some regulatory elements. And when there's no lactose present, do you get expression from this promoter? Is it turned on? No, because there's a protein called LAC repressor that sits on the operator, blocks polymerase, and so you get no expression. If you add lactose or a chemical that mimics lactose, so IPTG is a chemical we use in the laboratory, looks just like lactose, binds LAC repressor, takes it off of the operator. It's just much more stable. If you add that to the cell, you can cause high levels of transcription, but only under a particular situation, right? So normally you would get low levels of expression in less there's low levels of glucose, right? Because then you get high CAMP, you turn on CAP, and then stabilizes the polymerase to sit on that promoter sequence that's non-consensus. But if you want to design this system to pump out high levels of a transcript when you want to turn it on, can you just change that non-consensus to consensus sequence? Yeah, so you can change it to consensus, and what that means, glucose levels aren't going to matter anymore. All that's going to matter is if you can get that Lac repressor off of the promoter. So it allows you to very easily regulate expression from this vector. Okay, so now how can we use this to make a human protein in bacteria? So what you do is you remove the gene that normally is driven by the Lac promoter, and then you can insert, using your cloning methods, any coding gene you want. So the open reading frame of a protein of interest. The example here is this GCSF. This is used to increase the immunity in cancer patients. And the important thing to know here is you need to clone the open reading frame of the gene of interest. What do we mean by that? It only has the coding sequences in it. Why wouldn't we include the introns? Antonio? Yes, bacteria do not splice out the introns. So you just get the sequence that just has the open reading frame. So when that messenger RNA is produced, the bacteria will take that messenger RNA, use their ribosomes, the code is universal, and make the protein. All right, and so here's where that Lac promoter comes into use. So once we do our cloning, we can then put our recombinant DNA into bacteria where there is no lactose, there is no IPTG, and then we can grow up large amounts of this bacteria containing this recombinant DNA until we're ready to turn them on to make the protein. All we have to do there is we add IPTG to the cells, takes Lac repressor off of the promoter, we make tons of this messenger RNA, the bacteria make tons of this protein, and then we're able to isolate it and use it for medical purposes. This is done in bacteria cells, but you can have different kinds of regulatory promoters, and you can use it also in other different types of cell types. And in fact, this is how many of our medically relevant proteins are made and given to treat a wide variety of diseases. So if the protein can be used in a human disease, you just have to design the open reading frame for that protein, put it in your expression vector, regulate expression, isolate the protein, and you have it available to give to patients. Any questions? Yes, Madeline.
So I read where sometimes people are working in a lab and they're doing stuff like this and somehow it gets out or something. What happens to those scientists or whatever? Will they have to have something inserted to turn off the promoter or repress it?
So typically the cells, the bacteria that we use for these types of experiments have changes in them so that the cells cannot survive in the wild. So the E. coli strain we use can't infect humans. So yeah, it's important to have these safety precautions, definitely. All right, so now let's move on to the topic tonight, which is going to be genomics. And what we mean by genomics is that it is the genome-wide analysis of gene structure and expression. So now that we're able to do all this sequencing, we have tons of genomes available to us. And so how can we access them? How can they be useful? And so we'll talk about where you can find these sequences in freely available sequence databases. We'll talk about one algorithm, an online program where you can take a sequence that maybe you don't know anything about and ask, is it similar to any other sequence that's been studied before? And by doing that, you might learn something about this new sequence you have and maybe what its function is. We'll talk about how we actually identify the open reading frames when we're looking at large genomic DNA sequences. And then we'll talk about two methods whereby we can analyze the expression of thousands of genes all at the same time. So there's been many, many genomes that have been sequenced. Our own human genome is over 3 billion base pairs long. musculus, what is that? Very relevant laboratory model. It's mouse, exactly. So its genome's a little bit smaller than ours, and we've talked about Drosophila, C. elegans, Arabidopsis is a plant, this is yeast, and then of course this is bacteria, all various sized genomes. Oh my gosh, what is this? This Neoceratodus forster with a 43 billion base pair genome. Anyone know what that is? It's a lungfish. Why do you think its genome is so huge? Does it have that many more genes than us? What do you think it's actually filled up with? Introns, but what hop around the genome and multiply? Actually, our genome consists primarily of these two. Transposons, yes. So transposons have gone wild in this organism, and that's why its genome is so big. All right, so where are these genome sequences stored? There's a variety of places, but one is at NCBI, and so if you go to this site, you will see a page that looks like this, where you can search for the genome or sequences of your favorite organism. You can see here over 3 million genomes have actually been sequenced at this point. We're still behind at annotating them. Annotating means going through and saying, here's a protein coding region for this gene, here's this tRNA gene, here's this gene. So we need to know what the sequences are in the genome. And so now I want to take you through sort of how having all of these genome sequences from all trees, the whole tree of life, from all different variety of different organisms, can actually help us in understanding human genes that are involved in disease. So we're going to talk about this gene, NF1. It is the gene that is mutated in a disease called neurofibromatosis 1. And what happens in these patients is that they develop tumors in the peripheral nervous system. And so this, of course, is an extreme case where there's just cells dividing out of control. That's what makes up a tumor. And it's a pretty frequently inherited disease. And in fact, there's spontaneous mutations that happen in this gene as well. It's a very big gene. That's why it's susceptible to spontaneous mutations. So there were patients who had been diagnosed with this disease. You could see it was running in families. And so in the early 1990s, there was a lot of interest in trying to figure out, well, what is this human gene that's so important that when it gets mutated leads to this disease? And so at that time, pedigree analysis was done, and they were able to identify the messenger RNA sequence that seemed to be affected in these families of people showing this disease. And so they could go in and they could read the sequence of the messenger RNA. And so when they sequence the end of it, they read until finally you get a start code on your first ATG. What do we call that sequence before the first ATG? Yeah, it's the 5' UTR, exactly. And so you have the sequence and you just scan until you find that first ATG. And as soon as you find it, you've set the reading frame. And so you can use your genetic code to start identifying the protein sequence. So ATG, of course, is methionine. If you look at the code, GCC is an alanine, and so forth. All right, so we know the RNA sequence. We can translate it into a protein sequence. So this is where this BLAST program comes into play, because you know the protein sequence. We don't know what the function of this gene is, but maybe there is a gene that has been studied already that might have some similarity to this disease gene. And so that's where you can use BLAST. This is an online program. I'll show you how to access it in a moment. This stands for Basic Local Alignment Search Tool. So it's searching for alignment. So do amino acids in your query, that's the sequence you're searching with. Does it match anything that's already in the database? We would call that the subjects. And so it'll search for identical amino acids, but also give points for similar amino acids, right? Because in a protein, sometimes it doesn't have to be the same amino acid, but some are very similar to each other. And so it'll see how far you can get that alignment with identical or similar amino acids, and then it'll rank the subjects that it finds by p-values, and you know p-value is the probability that a degree of similarity is by chance. The lower the p-value, the more likely this is not happening by chance. And so typically we pay attention to results that are less than 10 to the minus third. So if you get a subject that comes up with your query, search, and blast that's less than 10 to the minus third, then it's likely those proteins are related. The lower the p-value, the more likely that they are related. So the way you can do this blast search is you go to this website through NIH, and it tells you this is the basic local alignment search tool. And there are various versions you can use depending on what your query sequence is. If you are searching with a nucleic acid sequence, like a DNA sequence or an RNA sequence, then you would do nucleotide blast. If you have translated it and you know the amino acid sequence, then you would do a protein blast. So we translated that NF1, we know the protein sequence, so we're going to do a protein blast. If you click on that, you will get this page. This is where you just would paste in your query sequence. And so I copied that whole amino acid sequence, pasted it in here. The other things to pay attention to are the database. So there's a variety of different databases you can search in. Most of the time, you're going to just use the non-redundant, this first option that's given up here. The other thing you might want to pay attention to, though, is organisms. So in this case, we want to ask any sequence that has ever been made, is that going to match to the sequence we're putting in? But if I had asked you, does this sequence have homology to anything in the C. elegans genome? Then here, under organism, you would put in C. elegans, and then it would only search that organism. So for now, we want to know anything that's in the database. And so then you just click blast here. And it'll come up and give you a whole list of things because there are many genes that have now been sequenced that we know have high sequence similarity to NF1. And of course, the very first one that comes up with identity, so 2,839 amino acids over 2,839 amino acids, is because the NF1 protein sequence is now in the database, right? Because I did this search like yesterday. Of course, your p-value, so when it says expect, that means the same thing as a p-value. The p-value is 0. There's no chance that these are not related. In fact, they are identical. It will show you your query sequence. So that's the sequence I pasted into that box. So that's on the top here. On the third line is the subject. This is what it pulled up out of the database trying to do the alignment. And then in the middle, it's just showing you where there were matches or similar amino acids. In this case, of course, every single thing matches. And so that's the result, of course, you get today. But when scientists did this back in the 1990s, the human genome hadn't been sequenced yet. There was no human sequence that came up. But luckily, there was a highly significant sequence that was pulled out by this blast search. And it was found in the Saccharomyces cerevisiae database. What is Saccharomyces? Yeast, yeah. So it's these single-cell yeast that we often use in the laboratory. It's long been used as a model eukaryotic cell. And so we can look up here, and we see that the p-value, or what they call expect, is 6 times 10 to the minus 53rd. That's highly significant. It's a very low p-value. And you can see where our query, where we put this in, compared to what it pulled out of the yeast database. All right, here, there's a leucine at both positions, so an L is put there. Then we have a difference in amino acid, and then we have the same. And then these pluses, that indicates where you have a similar but not identical amino acid. So you can see for this first plus, it's hard for me to see. We have a K, which is lysine, and we have an R, which is arginine. So those are two big positively charged amino acids. They're similar. That contributes to our score, giving us this very low p-value. And so it turned out that this human NF1 sequence had very high degree of similarity to a protein in yeast that had been called IRA. So, what was known about IRA? Well, it was already known that the yeast IRA is a GTPase accelerating protein that regulates the RAS protein to control cell division. So, some of you might remember we talked about RAS is this protein that, when it's active, promotes cell division. So that's important. Sometimes you want to have cell division, but it's also really important to regulate its activity. One of the ways it is regulated is through these GTPase activating proteins. So RAS itself is a weak enzyme. It is a GTPase. What does that mean? It means it binds to GTP and slowly hydrolyzes GTP to GDP. When it is in the GTP-bound form, that is the active or on state that's going to promote cell division. The way cells keep it off is there are proteins in the cell that are these GTPase activating proteins. So as the name suggests, they activate the GTPase activity of RAS so that it hydrolyzes GTP to GDP. This is the off state. You're not going to promote cell division in that state. It is reversible. There are GTP exchange factors in cells that can replace that GDP with GTP, put it back in the active state. So it's a very regulated form of telling the cell when to divide versus not. So if NF1 and IRA are a gap, a GTPase activating protein, can you now see how, if you don't have NF1, if you do not have this gap available, what's going to happen? RAS is going to be stuck in the on form. It's going to tell cells, divide, 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 divide. You get tumor growth. So by knowing what the function of this yeast protein was, it helped scientists very quickly identify why this mutation was leading to these types of tumors, and then how potentially they might be able to re-regulate RAS or maybe reintroduce these gaps to treat these types of patients. Now, sometimes there might not be a full sequence match to a protein that we put into the database and do a blast search for, but often there will be amino acid sequences, so just a small portion of the protein that can give you clues to its function. These are called structural or functional motifs. And they are just small segments of the protein that we know are associated with a particular function. So for example, we said RAS binds to GTP. It has a motif that allows the amino acids to hold on to GTP. And that motif is you either have an alanine or a glycine, and then you have 4 amino acids. That could be anything. Then you have a glycine, then you have a lysine, then you have a serine or a threonine. So if you see this motif in your protein and maybe nothing else, that at least starts to give you a clue. Maybe this protein binds to ATP or GTP, and you can start going further with experiments. And so many of these types of motifs have been identified, and you can look for them in your proteins once you know the amino acid. And so, I already answered the question for you. So RAS would have this GTP binding motif. All right, so let me just also take you through if you're going sort of in the other direction. So now we know that there's this really important gene, NF1, neurofibromin 1. So how would I be able to pull its sequence out of a database? So you can go to the sequence databases shown here, and you can put in the name of any gene, any protein that you want. You hit search, and it'll give you this menu of all different types of things you can look up. So PubMed, this tells you there's been over 2,000 papers written about NF1. It's a very important gene. It's a disease-related gene. You can also get the sequence for the gene, or you can get the protein sequence itself. And so look, there's over 6,000 protein sequences for NF1. Are those all in human? No, it's because yeast and everything in between has homologs of NF1. And so If you click on actually that link for protein, it will give you a potential list of all 6,000 of those different proteins. And at the very top, you'll see, so in parentheses here, these are the organisms. And so this megalopta, all right, what is that? It's a bumblebee, or it's not a bumblebee, but it's a type of bee. And then we get, this is a worm, and these are insects. Okay, so they all have a neurofibromin protein themselves, but maybe we are interested in looking at this protein sequence in humans. How do you easily get to that? You just go up here for your organisms, top organisms, humans usually the first one on the list, and so you can click on that. There's 90 human NF1 protein sequences. And so if you look at the list you get back here, what you'll notice is there's some differences in amino acid size. What do you think that tells you about this protein? Yes?
Has like different functions?
Not different, not necessarily. Different isoforms, right? So there's probably a lot of alternative splicing, and in fact, that is true. NF1 is a very complicated gene with many different isoforms, but there's not 90 different isoforms. A lot of these sequences are actually disease alleles. So they've sequenced from patients, they know there's a premature stop codon, or there's an amino acid change. So those could be included in here as well. To know exactly what you're looking at, so if you click on, for example, this one, that's 2839, that's the one we had looked at before, you get a page that looks like this, has all sorts of information, but it tells you the reference from where the sequence came from, and then if you scroll down that whole page to the bottom, there you go. There's the whole amino acid sequence of this isoform of NF1. So it's very powerful to have these freely accessible databases at your fingertips to look up any genes, disease-related genes you might be interested in. All right, so now let's talk about, we're able to sequence entire genomes, and once you have the genome, we want to annotate it, right? When we said annotate, that means figure out where all the genes are. Where's the protein coding gene? Where's the tRNA genes? And so how do we find these? Well, one of the ways we find the protein coding genes in simpler organisms with smaller genomes, like these yeast cerevisiae or an E. coli, is you can just scan for open reading frames. So you just scan through the genomic sequence until you find an ATG, and then in that reading frame, can you read for at least 100 codons, so make a protein that's at least 100 amino acids long? That is probably a protein coding gene. And so that is how much of the yeast genome was annotated. There's not a lot of spaces between the genes, and there's not many introns in yeast genes, so it's pretty easy to just start at the ATG, get to the stop codon, and that is the protein coding sequence. So that's good, but of course our own genome and genome of many other organisms is a lot more complicated than that. So just doing this ORF analysis, finding an ATG and starting to translate, is not going to work for the vast majority of genes because we have these huge introns that are interrupting our genes. If we just keep reading through there, we're not going to be in this right reading frame. We're going to hit stop codons pretty rapidly. And so what do we need to do in order to identify the protein coding sequences in more complex genomes? So one of the things, so we have algorithms that take advantage of all the data we can feed into it. And one of the pieces of data that's very useful is to say, okay, protein coding, proteins come from messenger RNAs. So what is the messenger RNA sequence, and then can we map that back to the genome to find where the exons must have been? So to do that, what we do is we align to what we call cDNA, or complementary DNA sequences. Complementary DNAs, or cDNAs, are just DNA copies of RNA. And of course, if you isolate RNA from the cell, the vast majority is going to be the mature spliced form. Introns are not going to be there because splicing is normally quite rapid. So what you can do is, if you want to identify all the copies of or all of the RNA in a cell and then be able to identify the protein coding genes. You take those messenger RNAs. We know messenger RNAs almost all end in a poly A tail, so we know its sequence is on the 3' end. We can then hybridize that to an oligo that consists of T's. T's will base pair to the A. We have a 3' end here. This is a DNA oligo base paired to RNA. We are going to use this RNA as a template to transcribe and copy the RNA into DNA. It's going to be a copy of the RNA. We're going to make cDNA. And what is the name of the enzyme we need to copy that RNA into DNA? A reverse transcriptase, exactly. So reverse transcriptase will copy the RNA. You then do a digestion to get rid of the RNA, and then there's various methods that can be used to make the second strand. I'm just going to tell you about one, but don't worry about so much how it can happen. So one way you can do it is that you can ligate a known sequence onto the 3' end here. So for example, if I ligate GGGGG, then I can make a DNA primer that will base pair to that sequence. 3' end is here, and now I would use a DNA-dependent DNA polymerase because it's going to copy DNA to make the second strand of DNA. Once you have your DNA, you can sequence that. And when you sequence it, what will happen is this might be how the genome looks, but this will be the sequence of your cDNA. Why does the cDNA miss the introns? It's a copy of the mature messenger RNA, exactly. And so what the computer will do is they'll recognize this sequence matches exactly here, this sequence matches here, this sequence matches here. And so it automatically tells you where the introns must be for that. And so by generating cDNAs, sequencing them, and mapping them back to the genome, this helps tell us, one, what sequences are transcribed in a cell, because we have a copy of the RNA, and then also, are there introns, or how do these map back to the genome? We also take advantage of knowing information about what intron sequences should look like, where do these really have splice sequences within them. And then we also can compare genomes across organisms and look for homology. So as you know, many protein coding genes in mouse, for example, actually have homologs in humans. And homolog is just defined as all genes that share enough sequence to suggest a common ancestor. So by using these methods, we've been able to annotate many genomes with the predicted protein coding genes. And so we know the human genome probably has between 20 and 25,000 different protein coding genes, and similar amounts in all these various organisms, quite a few less in yeast. By looking at homologs across organisms, and also for those motifs that tell us something about a function of a protein, we can classify some of these into what we think they might be doing in the cell. But the thing that should really stand out to you here is this light blue piece of the pie, where the vast majority of these predicted protein coding genes, we have no idea still what their function might be. And so there's a lot still to be discovered here. All right, so what we've been talking about is genomics, which is really the sequencing and studying of entire genomes. And one of the things that has really allowed us to be in what we consider this genomics era is this ability to do this next generation sequencing, this parallel sequencing. And again, if you go to this site, you can see visually how this takes place, but don't worry about the details of how next generation sequencing happens. Think more about why you would do it and what you can learn from it, because then you can apply it. And in fact, this of course has been applied. It's allowed us to sequence the genomes of millions of different samples. So we have genomes across the tree of life. But we also use this method in the laboratory for some protocols that we've already talked about in class before. So remember our DNase protection assay? So remember we wanted to do this to ask if a particular gene was in tight condensed chromatin and therefore repressed, or loose open chromatin. And we did this for the globin gene. But now we can ask not just one specific gene in our sample, but the entire genome. Which genes are tightly packed and therefore protected from DNase versus those that are loose and open. The way we would do this is we'd start exactly like we talked about previously in class by isolating the nuclei, treating with DNase. If it's packed up in chromatin, it's going to be resistant. If it's open, it's going to get degraded. You then extract the DNA. But then, instead of using restriction enzymes and doing your southern blot with a probe for your specific gene of interest, you just take all that DNA and do next generation sequencing. If it was in the protected region, you're going to get a lot of sequencing reads, right? There's going to be a lot of available DNA to sequence. If it was in the degraded region, you're not going to get reads that are going to map to the genome in that region. So through this, we're able to say in two different cell types, what are the differences in chromatin under these particular conditions by simply sequencing the entire genome. The other type of experiment we talked about where next generation sequencing is very powerful is the ChIP experiment. Remember, we wanted to use chromatin immunoprecipitation to ask if a specific DNA sequence was associated with a specific protein of interest. With next generation sequence, we can identify all the sequences in a cell that are associated with your protein of interest. For example, if you wanted to know all the sequences that are associated with RNA polymerase II, in your ChIP experiment, after you use an antibody to isolate RNA polymerase II and any DNA that was cross-linked to it, when you then release the DNA, you just sequence it. So instead of doing PCR for a specific gene, you sequence everything that was cross-linked to your RNA polymerase II, and you know everything, all the genes that were associated with your protein of interest under that condition. Does this make sense? All right, so again, genomics is sequencing and studying entire genomes. When we're talking about genomics, we're talking about DNA. We also have this term transcriptomics, and that is the ability to detect and study all transcripts. Transcripts are RNA. What is the RNA that is produced in a cell at a particular time? And we'll see how we are able to do that. And you may have heard of proteomics. Proteomics is looking at all the proteins that are present or that immunoprecipitate with a given protein of interest. And there's now also metabolomics. All right, so now let's talk about two general methods we use for transcriptomics to ask what RNAs are expressed and to what level in a particular cell type or under a particular condition. And the first method we'll talk about are microarrays. So these allow us to detect the expression of thousands of genes, thousands of different transcripts at the same time. The way they work is they take advantage of these little microarray slides where each square is going to have probes or oligos for a specific gene. And so gene A might have probes here, gene B here, gene C here. And so it's going to have many, many copies of oligos to which a cDNA can base pair to. So how does this method work? Let's say, for example, we wanted to ask how are RNAs differentially expressed in fibroblasts, skin cells that were growing in culture, either with serum, so nutrients, or without serum. Probably there's going to be a lot of changes in metabolism. There's going to be a lot of changes in gene expression. We want to detect all of them at the same time. So the first thing you need to do is prepare your microarray slide. So make sure that you have probes for all the genes you want to look at. These days, we buy them from companies. If these are human cells, we would buy a microarray that has probes for all the human genes, for example. You then isolate RNA from these two different populations of cells and then synthesize cDNA, but in a differentially labeled way. So we can add a dye that will make The cDNA, so all the copies of the mRNA in the fibroblast without serum, will be labeled with a green dye. And all the cDNA made from our fibroblasts that had serum will have a red dye. So we've differentially labeled our two populations. You can then mix them together, add them to the slide, and then you will get hybridization or pairing of the cDNA. So we keep the cDNA in the single-stranded form here. We don't need to make the second strand because we want it to be able to base pair to the oligos for each gene we have here. So if this spot for A that has probes for A lights up green, what that's telling me there is a lot of cDNA that was from the fibroblasts without serum that's available to base pair to that spot, making it glow green. B is glowing red, so we must have had a lot of cDNA made with cells without serum. And then gene C here, we're getting yellow. Green and red make yellow. So we have pretty similar levels of gene C being expressed under both different conditions. And so these little lines, that's just to illustrate that there's a whole bunch of DNA oligos of the same sequence there. They're stuck in that position. And so when you get pairing of the cDNA that has this fluorescent label, there is a very sophisticated microscope and computer that will detect that signal and then give you a readout. And so here's one way to visualize and interpret these results. So remember, every spot is going to be a different gene. You know what gene it is, and so you automatically get information back about the relative expression level. Everywhere we see a green spot, remember that in our minus serum conditions, that's where we use the green fluorescent to make the cDNA. That means we had high levels of expression of that gene in the minus serum conditions. Plus serum, it was red, so some genes come on only under the plus serum conditions. And so in this particular microarray, they had 8,600 different genes, and they found that 500 of them were showing either increased or decreased expression after serum addition. This is another way, just sort of written out again. So since we labeled the without serum with the green dye, that is telling us that if we see that spot glowing green, it means the mRNA is at higher levels in the minus serum conditions, or that it decreases when serum is added. It's the opposite for red. Yellow is if there's similar expression under both conditions. We're making cDNA from both cases. They are base pairing to the probes. And then black just means there was no cDNA to pair to those probes, so that gene's not expressed, right? All of our genes aren't expressed all the time, so you're going to see a lot of blank spaces. Now, of course, we don't just look at spots that are come up like this. We actually get quantitative numbers associated with all of this. And so we might see for gene A, there was no cDNA pairing to it. It's just blank, so that probably means there was no expression. We might, for B, see very bright green fluorescent, and the computer will tell us 100, probes or 100 cDNA were paired versus just one from the plus serum. So there is a very high level of expression under the minus serum conditions. We might see a little bit of red and green and yellow, so maybe slightly higher expression in plus serum compared to minus serum. We might see the same levels here, and then we might see a higher level here. So we're given back numbers, and usually it's fold difference. It's differential expression, because that's really the question that we are asking here. And so that's how we would do microarray analysis. It requires us to already know the gene sequences that we're interested in, because that needs to be on the array with the probes. In contrast to that, we also have now the ability to do RNA sequencing, where you don't necessarily need to know the sequence of the genes you might be looking for. So to do RNA sequencing, what you would start with, again, is two different conditions, your samples of interest. For example, maybe normal colon tissue versus a colon tumor. So what genes are differentially expressed in these two conditions? You isolate the RNA, and now we're eventually going to want to do next generation sequencing. So what do we need to do? We need to convert those RNA sequences into cDNA. Once we have DNA, we can do our parallel sequencing. So you add the linkers, put it on a flow cell, you get parallel sequencing, millions of reads happening all at once. You get those sequences back, you map them to the genome. This will tell you the more reads you get means the higher the level of that RNA. But also because they will be mapping to, based on the cDNA was made, you also know what exons were expressed or not, so you can detect alternative splicing with this method as well. So here's an example, sort of a cartoon example of what this might look like. So what you're looking at here, so sex lethal, that might ring a bell. Remember when we talked about alternative splicing? This was a gene expressed in Drosophila. Turns out, so these blue boxes are the exons. There's 10 different exons here. And we've looked at expression levels of sex lethal from 2 different samples here. Every time we get a read, so one of these lines means a read, and so it will map to the genome where it corresponds to. Why in both samples am I getting these blanks over the black lines? What are those? Those are the introns, right? So remember, we get very little intron sequence in our cDNA because there's rapid splicing. Another thing you really need to pay attention to is over here, see where it says reads, and then look at the scale. So 1 to 1,000 versus 1 to 100. So that's important to pay attention to. So with that in mind, which of these samples, 1 or 2, has the higher expression levels of sex lethal? 1, yes, yeah. So the scale is all the way to 1,000. We have much higher levels of reads than what we're seeing in sample 2. Does anyone notice another thing that we can identify by looking at where these sequencing reads are mapping? Any other difference between sample 1 and 2? Yes, Cleo?
Number 3.
Ah, yes. Exon 3 is here in sample 2, but where'd it go in sample 1? It was alternatively spliced. It was not present in the messenger RNA, so when you copied and made the cDNA, that sequence was missing. So we can learn about the level, relative level of RNA expression. Also, if there's alternative splicing. We also can learn about alternative transcription start and polyadenylation sites, right? So if transcription started down here, we wouldn't get any reads at that position. If polyadenylation stopped at a different position, we wouldn't get reads beyond that point. So there's a lot of information you can get by doing RNA sequencing. Question?
I didn't capture what you said why we have like 100 and there's 1,000 reads. What was the
So this is the scale. So 1 to 1,000 meaning this means that we have 1,000 reads kind of piling up at this position. Here, the highest we have is about here, so that's maybe 25 if we're looking at the scale here. So it tells us the level of reads. Many of these reads map on top of each other, so you can't just like count which. And this is a cartoon version. I'll show you what this really looks like in a moment. Based on maybe what you remember about sex lethal and which the XX or the XY makes this, what do you think sample 1 is? The female, exactly. So remember, female is able to skip exon 3, which means it doesn't undergo nonsense-mediated decay, which is what's keeping the levels so low here in this sample. All right, let's finish off then with a clicker question based on interpreting RNA sequencing reads. So which of the following conclusions is consistent with the below RNA-seq reads of gene X? So what we're looking at, we've done RNA sequencing of gene X in young versus old worms. Here's the scale. And this is actually what those reads look like. And so it's not those nice, like, very defined lines, but you actually get these peaks piling up on each other. Nick. Yes, absolutely. All right, 10 more seconds. All right, final answers. All right, so we have a lot liking E, more than one of the above. And so I think rather than rush through all of these, what we'll do is I'll keep you in suspense until Tuesday. We'll start class with this question again and then go through each one. Have a great weekend.